So in this lecture, we're going to focus on species, interactions, and community structure. So this is the material in chapter 17 of your textbooks. So we're going to start by describing some ecological networks, focusing mainly on trophic networks or food webs. And then we're going to look at some of the factors that drive community assembly. So what are the factors that determine what species are present in a community? And two of the big factors that we'll touch on are competition and the activity of keystone species. An ecological network is a description of the patterns of species interactions in a community. And there are three main types: trophic networks, host parasitoid networks, and mutualistic networks. Trophic networks describe interactions based on feeding relationships, so these are food webs. Host parasitoid networks describe the interconnectedness of host species and their parasites, diseases, and parasitoids. And mutualistic networks focus on interactions like pollination and mycorrhizae. The most familiar form of ecological network is a food web, so a form of trophic network. So this is just a description of feeding relationships among species in a community. And different ecological interactions can result in predictable assembly rules that govern the structure of communities. So assembly rules are an idea that was originally something that Jared Diamond proposed. And they're really useful things because they're things that we can observe in a community or interactions that we can observe that allow us to make predictions about the structure of communities. And although very useful, there's a lot of debate around assembly rules. Are there any that work particularly well? So we'll have a look at some examples of that as the lecture goes on. So in terms of ecological networks, we're going to focus on food webs. So food webs describe the feeding relationships in communities. And one of the first really well constructed ones was developed by Elton and Summerhays. And they chose a location for their food web, Bear Island, which is in the Arctic Circle off the northern coast of Norway. And they chose that site because they thought there were relatively few species there. This is a very kind of remote and extreme climate. And so they created this food web that essentially shows primary producers being small terrestrial and aquatic plants and algae. And those are fed on by various insects, which in turn are fed on by birds. And then those are fed on by Arctic foxes. And there's also another large mammalian predator, the polar bear, which feeds mainly on the carcasses of washed up seals and other marine mammals. So these food webs are really useful because they allow us to visualize these feeding relationships in a community, but they're only showing us trophic interactions or feeding relationships. And that's only really one aspect of how a community works. And even though they're only mapping this one type of interaction, there's growing kind of recognition that food webs tend to do this incredibly badly, or at least in an incomplete way. So what I mean by that is there are Elton and Summerhays food web shows insects feeding on aquatic and terrestrial plants, but they only specifically mentioned 3 or 4 different insects, where in fact there are hundreds, potentially thousands of insects that do that feeding. And this incompleteness in food webs tends to follow a particular pattern as well. So the larger, more characteristic animals like the Arctic fox and the polar bear, they're obviously all included, but as you move down the trophic levels, you get more and more incomplete pictures and incomplete data sets. And there are practical reasons why ecologists might focus on larger animals when they're studying feeding relationships, and we'll come on to that when we talk about some of the actual techniques of data collection that these ecologists can use later on in the lecture. But the truth is, if we want to understand this community, understanding the role of Arctic foxes and polar bears is perhaps no more important than understanding the role of some of these insect species or microbes operating further down the trophic scale. And another thing that was absent from Elton and Summerhays' initial food web was the activity of decomposers and detritivores and the effects of soil organic matter. So these things that are more to do with nutrient cycling and lower down the food chain tend to be omitted from many food webs. So another important ecologist when it comes to constructing food webs was Ralph Bird, who in the 1930s constructed food webs for different communities within the aspen parkland in central Canada. So he looked at prairie communities, aspen forests, and willow stands. And he constructed food webs for each of those. And you can see two of those on the left-hand side of this slide. The far left one is prairies, and the middle one is the willow communities. But what was really important about Ralph Bird's work was that he was one of the first people to acknowledge that community boundaries are fuzzy, they're not distinct. And so there is this exchange of energy between communities. And you can see on his food web on the right-hand side, he's got the 4 different communities that he studied and connections between them. So he acknowledges that there are direct interactions of species among communities. Bird's food webs, though, showed a similar pattern of incompleteness. So you can see on the left-hand side, insects are grouped pretty broadly into orders, but many of the birds he observed are listed down to species. So this is likely just down to his greater interest in birds, or his assumption that those birds are more important. But of course that's not necessarily the case. It should be noted though that although a weakness of many food webs is their incompleteness, that does give them a degree of utility that maybe more complete food webs wouldn't. And so this food web on the left-hand side, comically described as a simplified food web of the Northwest Atlantic, shows how quickly these food webs can become too complicated to be of any use, really. And so this is a reason why many food webs are kept simple and kept incomplete. For the next few slides, we're going to look at the question of how competition between species can affect community structure. And the answer to that question really depends on whether species have similar competitive abilities or whether the community contains competitive hierarchies. So if a community has many species which are equivalent in their competitive abilities, then in theory many of those species should be able to coexist. But if there's a competitive hierarchy, some species are better competitors than others, and some of those less competitive species are likely to be excluded from the community. So the simplest example of a competitive hierarchy would be a community with 4 species, A, B, C, and D, and those species can be ranked like this. So A is the most competitive, then species B, then species C, and then species D. Most competitive hierarchies, though, would usually be more complex, and one such slightly more complex competitive hierarchy is displayed in this diagram. So species A is the most competitive here. Species B and species D are equally competitive, so they can both outcompete species C, which is the least competitive species. As the level of competition, so the intensity of competition in a community increases, it's more and more likely that some of these less competitive species will be excluded from the community. Another factor that affects the likelihood of that is whether competitive hierarchies are transitive. So what do we mean by transitive? Well, this is a term really from basic algebra. So if you look at that really simple competitive hierarchy where A is stronger than B, is stronger than C, is stronger than D, then if it's transitive, that means that species A is also able to outcompete species D. So it's essentially just, as I say, kind of an algebra term. And so if that's the case in a community, as the strength of competition increases, slowly we'll lose those least competitive species. So we'll lose species D, lose species C, and then eventually that community might be dominated just by the most competitive species, species A. So the theory then is that when multiple species, which are functionally similar and competing for the same resources, when they co-occur, the more competitive species will outcompete and exclude the less competitive species. And this is an example of an assembly rule, something we can use to make predictions about ultimately the makeup and structure of communities. But many ecologists have worked hard to try and establish whether these rules actually hold true in natural systems. So an example of some of these ecologists are Paul Keddy and Irene Wishow, who've worked for a long time in freshwater wetland habitats of Ontario. And one of their early observations was that the kind of core parts of these wetland habitats are dominated by one type of grass, typha. So that's the picture you can see on the left. But in kind of the slightly peripheral parts of the habitat, there are areas that contain a diversity of other species. And so they use these observations to develop their concept of centrifugal organization of plant communities. So the concept of centrifugal organization of species is based on the idea that any landscape is divided into core and peripheral habitats. So in those core habitats, you have abundant resources and very few abiotic or biotic stresses. So you have probably lots of sunlight, low salinity, low numbers of pests and pathogens. And in those core habitats, the species that thrive are the best competitors. So the species that can utilize those available resources most efficiently are the ones that survive and dominate in those core habitats. So in contrast to the core, there are many peripheral habitats which differ from the core in various different ways. So they have greater abiotic stress, so maybe less sunlight, higher levels of salinity, and greater biotic stress as well. There's probably more pests, more pathogens, so other things that species have to tolerate. And in these peripheral habitats lie many species that have probably low competitive ability, but greater ability to tolerate stress. So what centrifugal organization of species really does is combines competition theory with allocation theory. It's really describing a trade-off between competitive ability and the ability to tolerate stress. And that's depicted really well in this figure, which shows comparisons of fundamental and realized niches in many of these species. So in the top competitor, typhus in this case, the fundamental niche is actually pretty narrow, but these plants are very competitive in these areas, and so they tend to be abundant there. As you move down to less and less competitive species, their fundamental niches are really wide. So this means they could occur in a really wide range of conditions, but of course they don't because they're restricted to their realized niche by competition. Typhus, on the other hand, is restricted to its narrow fundamental niche by abiotic and biotic stress. So let's focus now on the influence of keystone species on community structure. So a keystone species is any species that has disproportionately strong influences on community structure. And the idea of keystone species was proposed by Robert Payne in 1966. And his idea was based on his observations of predators. So he saw that predators can keep prey species below carrying capacity. And when prey species are below carrying capacity, they're unlikely to competitively exclude other species because they're not utilizing the available resources to their full capacity. And so any predator species that can kind of decrease the likelihood of competitive exclusion within a community is thereby increasing species diversity in that community. And so Robert Payne proposed the idea that many predators may be keystone species. So Payne put this idea to the test by studying rocky shore or intertidal zone communities in the northern US. So he identified a community where he wanted to work, where he found one apex predator, which was a sea star, a middle level predator, which was a snail species, and then 13 prey species that he observed in these communities. So they were made up by things like chitons, limpets, bivalves like mussels, and barnacles. So what Payne did was in plots on these beaches or in this intertidal zone, he removed that apex predator from the community and then watched how the community changed in response to removing that predator. What he found was that after 1 year of observing these communities in the absence of sea stars, the number of prey species had reduced from 13 to 6. So a huge drop in diversity over a relatively short space of time. So after that year, the community was dominated by a couple of mussel species and 1 barnacle species, so much greater level of domination by a few species. And it seemed to be largely space that these species were competing for. So in addition to lowering the number of these prey species, algae also declined because of the lack of space, and sponges were also crowded out. So Payne here was really vindicated that this, the presence of this sea star, so the apex predator, even though it occurs in relatively low numbers, has a huge impact on the structure of the community beneath it. So another good example of the activity of a keystone species comes from Eel River in northern California. And this river is characterized by a cycling of algal populations. So there tends to be kind of dense mats of algae on the riverbed in the wet season, which is the winter. And this algae then dies back over the summer. So you can see the picture from August shows very little algae on the bed of the river. So the food web in this system looks something like this. You get the algae at the base of the food web, and that's fed on largely by midge larvae called chironomids. And those chironomids are fed on by predatory insects and small fish, and then those in turn are fed on by large roachfish and steelhead trout. So the question that Mary Power in 1990 asked was, do these large fish have a significant effect on the food web structure? So what Power did was she developed plots within the river from which she excluded these large fish, so the large roachfish and the steelhead trout. And she wanted to know whether the exclusion of those fish, or what effect the exclusion of those fish would have on the structure of the food web. And what she found was that in the plots where those large fish had been removed, she saw this huge increase in algal biomass. So what seems to be happening is that when you remove the large predatory fish, you get a huge increase in the populations of small predatory fish and predatory insects. And those increased populations are then feeding more and more on the chironomids. So the population of chironomids has gone right down, and so there's very little herbivory on the algae, and so as a result you're getting this huge increase in the algal biomass. So these large predatory fish are having an indirect effect on species several trophic levels below them. And this is an example of a trophic cascade, this idea that if you remove, say, an apex predator, that's going to have an impact at every trophic level below that species. So over time, the idea of keystone species is something that perhaps became overused in ecology. So people using the term keystone species just for any species that they deem to be important. And so over time, we've refined the definition of keystone species to species whose impact on communities is disproportionate to their abundance, either in number or biomass. So in other words, a keystone species is not a dominant species. So there are many species like large trees that might dominate forest communities. And these species have a huge impact on their community by virtue of their biomass. But this is not an example of a keystone species. A keystone species is one with relatively low number or biomass, but that can have a disproportionate effect on the structure of the community. So things like the sea stars or the relatively uncommon large predatory fish that we've shown you over the last couple of slides. So in the study of keystone species, in the past we've tended to focus mainly on predators, and that goes all the way back to Robert Payne, who kind of coined the term keystone species. But increasingly we're finding that many mutualistic species also meet the requirements. In other words, they have low biomass, but a disproportionately high impact on community structure. So one good example of a mutualistic keystone species are seed dispersing ants in South African Fimbos. So native ants in these areas are responsible for 30% of seed dispersal in these habitats. And the seeds of many of these plants have structures called a lyosome, which are very kind of fat and lipid-rich structures that provide food as a reward for these ants. So native ants bury the seeds in their nests, and this keeps the seeds safe from many rodents who feed on the seeds, but also from fires. So fires are common in these habitats, and the seeds often are the only life stage of many of these plants that survive the fire. So ant dispersal of these seeds is critical to the survival of many of those plant species. Unfortunately, what's happening in these habitats is increasingly we're seeing invasions of the Argentine ant, which are a species that are invasive in many parts of the world. And these invasive ants are displacing many of the native ant species that disperse these seeds. The Argentine ants, incidentally, don't disperse any seeds. So these Argentine ants are displacing ant species that would usually disperse seeds. And in particular, it seems that plants with large seeds are being affected more by this. So the Argentine ants are displacing many of the ant species that would disperse large seeds. So this has resulted in a huge reduction in recruitment of shrubs producing large seeds in these habitats. The plants that produce smaller seeds probably have many ant species that still occur there, or they might have different dispersers. But this is a good example of how an organism with relatively low abundance, so these seed dispersing ants, can have such a huge impact on the makeup of their communities. So we've talked a lot in today's lecture about food webs, and I wanted to finish by kind of focusing on some of the tools that ecologists use to construct these food webs. So how do they generate the data sets on which these food webs are based? So one method that's been used much across the years is gut content analysis. So this picture shows a lionfish caught off the coast of Miami, and in its stomach the researchers found one shrimp and over 60 small fish. So we use this data to understand, first of all, the species that lionfish might eat, but also the level of impact they might have on prey species with which they co-occur. So this is a really useful tool, but it also does or has contributed to this bias towards studying large charismatic species. So for example, how would you do similar work if you were studying zooplankton or even marine invertebrates for which you wouldn't be able to identify what they'd eaten by cutting them open? Increasingly though, scientists have been able to use molecular techniques to conduct studies like this on much smaller organisms. So using DNA as a tool to understand feeding relationships within communities. And one example of this actually comes from work that I've been doing over the last few years. So until recently I was in South Florida working on the control of an invasive weed there called Lygodium microphyllum. So this is the weed here, and you can see it massively reduces native biodiversity by just kind of blanketing and smothering native vegetation. This weed's really difficult to control with things like herbicides, because although herbicides will kill the above ground parts of the plant, they leave behind these kind of woody mats of stems that make it very difficult for native plants to actually recolonize that space. And because the Lygodium grows from an underground rhizome, which isn't affected by the herbicides, it's usually the Lygodium that sprouts back from the herbicide use first. And so actually using these herbicides, which also kill other plants in the communities, might actually be selecting for, over time, a more serious Lygodium infestation. So the best hope for long-term sustainable control of Lygodium is biological control. And so I've been involved in a classical biological control program. So this is where scientists went back to the native range of Lygodium to try and find herbivores that we could then bring into the invasive range of Lygodium to try and control the plant there. So Lygodium originally is from Australia and parts of Southeast Asia. And so we went to those parts of the world to identify herbivores that we might be able to use as biological control agents against Lygodium. So many potential herbivores were found, but obviously you have to do a lot of host range testing and quarantine testing to make sure these insects aren't going to have non-host effects or non-target effects in their introduced range. And so we have a couple of insects now that we're licensed to release in South Florida, and the most promising one we think is this one. It's a small crambid moth called Neomusotima conspurcatalis. So it's only small, about the size of your fingernail, and females lay about 30 to 40 eggs that look like these kind of shingles. And they hatch out into these kind of almost translucent caterpillars that initially kind of just window the leaves of the plant and then eventually feed on leaf margins like this. But this is about as big as the caterpillars get. This one's about 1 centimetre long. But if we can get them established on the Lygodium in high enough numbers, they do this really impressive damage where they cause these brownouts or these big kind of dieback events in the Lygodium. So this picture comes from some helicopter monitoring that we did in a Lygodium infested site. And it shows these big areas of brownouts where the Lygodium has died back in response to these Neomusotima conspurcatalis moths. So I'll just call them Neo from now on. So this is an example of that biological control agent working really well. But we've actually been releasing this moth since 2008, and we're finding that this level of control of the Lygodium is something we see very rarely. So I did my postdoc work on this project, and a big question that I was asking or trying to address during that time was, what are the factors that are limiting the success of Neo? So why aren't these moths becoming more established more quickly, and why aren't they more effectively controlling the Lygodium? And it seemed to me that a big reason for that was likely the presence of lots of very voracious native predators. So we were going and releasing these caterpillars in Lygodium infested sites, and we'd release the caterpillars in their thousands, but the caterpillars had seemingly very little in the way of defense against predators. And so it seemed likely that some native predators were kind of treating these releases as a bit of a buffet and kind of removing large proportions of these Neo. So it became important for me to understand what are the insects that are most commonly eating these Neo. And if we understood that, then we might be able to change the way we release these Neo in order to get them better established and provide better control of the Lygodium. Unfortunately, because Lygodium grows in these kind of really thick mats, and often the sites where we were releasing these Neo were quite inhospitable places. We're in the middle of the Everglades, often waist deep in water. It's very difficult to actually directly observe these insect interactions. And so although we suspected that many native predators like ants and other predatory insects were reducing the populations of these Neo, those interactions were more or less impossible to actually observe in the field in real time. So I needed to come up with a way to determine what those interactions were, so what was most commonly feeding on the Neo, but without being able to observe it firsthand. And what I decided to use in the end was a process called DNA barcoding. So for DNA barcoding, what we needed to do was identify a stretch of DNA that was completely unique to Neo. So we used some of the mitochondrial DNA and ultimately found a stretch of DNA about 600 base pairs long that was completely unique to Neo, so it wouldn't be found in any other species. So what this figure does, although it would be very difficult to make anything out of it, it just compares that stretch of DNA to the equivalent stretch of DNA in some very closely related moth species. So it just confirms that we found a completely unique sequence of DNA in these Neo. So that was our DNA barcode. So having identified this DNA barcode for the Neo, so the biological control agent, what we could do is bring predatory insects from our field sites into the lab and just deliberately feed those predators on Neo. So then, once we knew the predators had fed on Neo, we could store those predator samples individually on ethanol and freeze them, and eventually run them through a PCR analysis to see if we could actually detect the remains of that Neo in the guts of the predators that we'd fed on them. So this is an early kind of electrophoresis gel that we used, and anywhere where you can see these white bands, this is a positive for Neo DNA in those predator samples. So we'd shown pretty quickly in a broad taxonomic range of predators, so ants, coccinellids, lacewings and spiders, that if those predatory insects had fed on Neo, we could identify that or clarify that using this molecular technique. So what that then meant was that we could go out into the field where we release these Neo and collect predators from those release sites. So we'd release several thousand Neo in a site infested with Lygodium, then come back several days later and collect large numbers of predators from those sites. So we could use these things called beat sheets that you can see in the picture, where we actually shake the mats of Lygodium and catch any predatory insects that fall out. So then we aspirate them and store them individually on ethanol and ultimately run them through these same PCR reactions. And what we found pretty quickly was that there were lots of predatory insects feeding on Neo in the field. So several ant species, several bug species, so hemipterans, several spider species. But what we found more than anything else were two species of carpenter ants. This is Camponotus sexcitatus on the left and Camponotus floridanus on the right. These are really commonly occurring predatory ant species in South Florida habitats. And these were the species more than anything else that were showing us positive for having fed on Neo. And what this actually does for us is that it allows us to maybe, or it informs our release strategies when it comes to releasing these Neo. So we can think, are there certain habitats where we can release Neo where they're less likely to come across these predator species? Are there different times of year that we could release Neo where they'd be less badly affected by these particular predators? So what this does is it gives us lots of information that we can use to improve release strategies and hopefully make sure that Neo becomes established more successfully and becomes more efficient or effective in controlling Lygodium.